So let's start with our first speaker. So our first speaker is Cameron Hassall from the University of Oxford. He's a postdoc in the Lawrence Hunt Lab, and his talk is titled Regression-based EEG analysis reveals temporally scaled signals during interval timing. So Cameron, the floor is yours.
Well, thank you very much for that introduction, and thanks everyone for coming to my talk. I'm very pleased to present today results showing temporally scaled signals in EEG. This work was done in collaboration with Jack Harley, a former master's student, and was supervised by Niels Koling and Lawrence Hunt. So action and perception in the real world require flexible timing. For example, the same piece of music can be played quickly, presto, or slowly, largo, and everything in between. We can also recognize the same piece of music played at different tempos. But how do we do this? Several models of timing have been proposed, and one in particular focuses on our ability to produce the same action over different timescales. That model is temporal scaling, which arises out of a recent animal work examining neural activity during interval timing. On the left here, you can see a monkey that is cued to produce either a short or long interval. On the right, you can see the response profiles of several neurons. Most of the recorded neurons in medial frontal cortex scaled their response to the duration of the timed interval. That is, the same response profile was stretched or compressed, depending on the interval to be timed. Contrast this with fixed time components, that is, activity that occurs over a fixed time scale, like these responses over here. The authors concluded that flexible motor timing relies on the speeding up or slowing down of a common network within MFC. Our goal was to determine whether or not these fixed time and scaled time responses could be dissociated at the scalp. This is challenging for two reasons. First, activity in the cortex is mixed at the scalp. A conventional EEG analysis, the method of event-related potentials, or ERPs, can only account for fixed time components. What I mean by that is that conventional averaging procedures assume that neural activity progresses over a fixed timescale. For example, you can see response-locked ERPs related to interval timing. Differences are apparent in the ramping activity, however, these signals share the same fixed timescale. Thus, it is unclear whether they contain any scaled components. In order to explore unmixing procedures, we simulated EEG in an interval timing task. In this task, participants see a cue, wait for a target duration, then respond at one of three target times, either short, medium, or long. We simulated two fixed time components, one for the cue, one for the response. We also simulated a scaled time component which spanned the delay period between cue and response. We summed these components together with some noise, and hopefully you can see in the resulting signal, components are now mixed. To illustrate what the results might look like in a conventional ERP analysis, we constructed cue-locked and response-locked averages, shown on the right. As you can see, adding in a scaled time component resulted in ERP differences that are consistent with previous literature, that is, different degrees of ramping activity. Our goal, however, was to unmix scaled time and fixed time components in order to recover the ground truth signals. So how did we do that? We used a regression-based approach in which the continuous EEG is modeled as a linear combination of underlying components. In our case, time-lagged regressors represented the contribution of our signals around each event of interest. These are variables or stick functions, that is, ones at the time when the component contributes and zeros otherwise. For much more information on this approach, I recommend checking out the Unfold toolbox. As you can see, we've included two fixed time regressors, one for the cue and one for the response. We then added a scaled time regressor whose contributions spanned the interval between cue and response. The idea here is to estimate a single component that accounts for the delay activity across a variety of timed intervals. And one thing to note is that we can solve this equation at multiple sensors. Thus, it might be possible to generate a scaled time topography. We then applied this new regression approach to our simulated data, and we were able to recover the original components, that is, fixed time components for cue and response, and a single scaled time component. You'll notice that the horizontal axis of the scaled component is not time per se, but rather the proportion of the interval. That's because this scaled component is spanning over multiple durations. So with this technique in mind, we conducted an interval timing experiment and collected some real EEG. In our temporal production task, participants learned to produce one of 3 target intervals, short, medium, or long. Blocks were preceded by a metronome to indicate the target interval. In the temporal judgment task, participants listened to a computer-produced interval, then judged the interval as correct or not. In the production task, we will be modeling fixed time components for the cue and the response, and a scaled component spanning the delay period. Likewise, in the judgment task, we'll be modeling the cue and probe as fixed components, and then a third component that spans this the gap between them. These plots show that participants were able to do the tasks quite well. They came close to hitting their targets in the production task, as indicated by the mean response times on the left. They were also able to correctly judge computer-produced interval, as indicated by the mean likelihood of making a correct judgment on the right. To align our results with previous EEG work, we constructed conventional ERPs, which interval-dependent ramping activity locked to the end of the interval, locked to the response. But these are fixed time components. Our regression-based approach revealed a scaled time component in both tasks that was distinct from the preceding and following fixed time components. Thus, we have successfully unmixed scaled time and fixed time components at the scalp. The signal appears to be consistent across tasks, both in the shape of the waveform and in the scalp topographies. And note that as before, the horizontal axis for our scaled component is not in absolute time, but rather relative time. That is the proportion of the interval that has been covered. We further tested our analysis procedure on open data from this paper by Breska and Duell, in which they examined rhythm-based expectations. The goal of their participants was to respond to a visual target that appeared sometime after a visual cue. When we included both the warning light and the target light as fixed time components and a scaled component as before, we again revealed a temporally scaled signal. This was a successful test of our method because although the fixed time components differed from our task, that is visual versus auditory, the scaled time component was remarkably similar in shape and location. So one question we had was, how does this temporally scaled signal relate to behavior? To answer this question, we used principal components analysis, or PCA. You may have heard of PCA as a dimensionality reduction technique, but what you might not know is that principal components often represent the temporal derivatives of a signal and may be related to component latency. This appeared to be the case with our data. When we ran a PCA on epoch data, the first principal component, PC1, matched the shape of our scaled time signal, shown in green. The second principal component, PC2, resembled the temporal derivative of this signal, which is supposedly related to the latency of the overall signal. To confirm this, we basically added in more or less of PC2 to PC1, so we sort of reconstructed the data in order to see what adding more or less of PC2 does to the signal. And indeed, it appears to shift the latency of the signal earlier or later in time. Thus, we considered PC2 scores to be a trial-by-trial index of component latency. We then related PC2 scores to trial reaction time data by binning responses as early, on time, or late within each of our conditions. Collapsing across conditions, we see that earlier responses tended to have greater PC2 scores compared to later responses. Thus, the peak or the latency of our scale time component was related to response time. The earlier it peaks, the earlier you respond. In conclusion, we've shown that temporally scaled signals can be recovered via regression-based analysis. In particular, we were able to unmix fixed time from scaled time components in EEG. In the future, we think it would be exciting to look for scaled time components associated with other processes that are known to unfold over different or varying timescales, including sequential actions, such as in music or speech, as well as decision-making, for example, evidence accumulation. So I would like to end by thanking my collaborators on the project, as well as all the agencies who funded the work, and a big thanks to all the organizers of Neuromatch. And thanks for your attention.
Thank you very much, Cameron. Yes, so if you have questions, put them in the Q&A window. So there's one question by Francesco Giosi, who asks, he's just asking for a paper to that references to this work. I think Alexandra has already put some information for that in the chat window.
Yeah, it's brand new work, so no paper or preprint yet, but stay tuned for that.
Okay. Do we have more questions? There's another one. It always takes a bit of time to type them. So Dongil Chang says, very interesting talk. Did you have a chance to check if the same framework being applied to decision-making problems where indivs may get to solve in different timing, or where individuals may get to solve in different timing, I guess?
Would you mind repeating that question?
Yeah, so I think the question is if you've applied your framework to decision-making problems,
right?
Where they solve problems with different timing.
Yeah, no, that I mentioned that at the end, that we're interested in this. Our lab also does work with evidence accumulation using a similar regression-based framework. So far, we haven't looked for scaled components within that framework. We're still working with fixed time components. But one of the sort of motivations for this work, when you look at these traditional ERPs, you can sort of see that one possibility is that these signals are actually being scaled. So you have a hunch that there might be some scaling going on there. So we definitely see a sssimilar type of activity in evidence accumulation.
Okay, and one last short question by Joe Arahua. Did you see any differences in your results for the regression analysis depending on the type of referencing used, especially in the topographic maps?
Good question. I mean, I would expect that as with conventional analysis, use of choice of reference is going to impact the topographies. I haven't actually explicitly looked at that, so we use an average mastoid reference to be in line with where they're looking at these ramping signals. But certainly is something that would be interesting to add, just to kind of confirm that, yeah, we are dealing with something that is like a regular topography and sort of behaves like a regular topography. Question.
Okay, well, thanks again, Cameron, for this very nice talk. And we'll move on to our next speaker.